In prokaryotes, transcription will immediately lead to translation. So in prokaryotes, the DNA goes immediately to an mRNA, and that mRNA will be immediately read to make protein. But in eukaryotes, there's the additional step. We first make a pre-mRNA, which will then be processed into a mature mRNA, and only the mature mRNA can be translated into a protein. So we're going to talk now about this processing step in eukaryotes. The pre-mRNA in eukaryotes is longer, and that's one of the biggest differences. The pre-mRNA is longer than the mature mRNA. The reason that it is longer is because it actually includes two things, introns and exons. Introns are material that was in the DNA and was transcribed into RNA. So it's in the DNA and it makes it into the pre-mRNA. But exons are things that will make it into the mature mRNA, and so therefore will be used to make protein. So introns are actually removed. Introns don't stay in. Exons do stay in because exons are expressed. The way that we get introns out of the pre-mRNA is through a process called splicing, which is the removal of introns. During processing, we also have the addition of a tail and the addition of a cap. So this is the pre-mRNA. Or rather, sorry, this is the DNA. So notice the promoter, the terminator, and the gene sequence, or in the transcribed region. The transcribed region includes both exons and introns, and so the pre-mRNA will also include introns and exons in the original mRNA. And notice that it's pretty long. It's as long as the DNA. Splicing is going to remove the introns. So the introns in this picture are green. So these introns will be removed, and then the exons, which are in yellow, will be put back together. In addition to splicing, a cap at the 5' end will be added, and a tail at the 3' end will be added. So we'll take a little bit of a closer look at splicing. Splicing is carried out by the spliceosome. And what the spliceosome does is it takes the mRNA, so I'm going to try and hold this up for you here. It's going to take the mRNA, and my hand is the spliceosome. It makes a loop, and then it has scissors that are going to cut that loop out. So they remove that loop, and then they put the exons back together. So you can see that in the picture there. It makes a loop, and then it cuts the loop out. The loop is green. So that's the intron, making a loop out of the intron, cutting it across here, and then leaving the exons to be put back together. So one of the things that's really fascinating about splicing is kind of the why, right? Why would we have introns if we're just going to cut them out when we make the mature mRNA? And one of the key reasons that this may have evolved is alternative splicing. So let me show you what alternative splicing is. So imagine I have a gene, and I have, so let me cord out of the way here. I have, this is my gene, and let me just use the same colors. So I have some exons in here. So I have exon 1, exon 2, and exon 3, and the rest is introns. So if I make a pre-mRNA with that, so let me change colors. That's the DNA, write that there. And I'm going to make a pre-mRNA that has all of those things as well. So, and again, I'll just use the green for exon 1, exon 2, exon 3. And then I'm going to make a protein. So imagine that I make a protein out of this. And I'm actually going to be able to do two different things because I can take different exons and combine them in unique ways. So I can take, oops, actually I'm one step ahead of myself, I'm sorry. And I'm also running out of room. So I'm not to the protein yet because of course I need to make a mature mRNA first. So I need to splice out my introns and leave in my exons and make a mature mRNA. When I do that, I can actually take different exons and combine them. So imagine I might have one mature mRNA that has exon 1 and exon 3 in it. I might have a mature mRNA with exon 1, 2, and 3 in it. I might have a mature mRNA that's only 2 and 3. So from the same pre-mRNA, from the same gene, I could actually make 3 different mature mRNAs, and therefore I could make, this is where I got a little ahead of myself, 3 different proteins. So each one of these would make a different and unique protein. So one gene can actually lead to different proteins. And this may have been really important in eukaryotes because we have lots and lots of information in our DNA, but we have even more proteins. So it may be a way to have a lot of diversity of proteins without having to have sort of an ever-increasing amount of DNA. DNA is difficult to take care of, it's prone to damage, so it's better to have less DNA and be able to use it to make more things. So again, I have one gene in the DNA, but I can use it to make 3 proteins. And that's alternative splicing. So splicing, again, is one part of RNA processing. The last 2 parts of RNA processing are the addition of a 5' cap. So that's at the 5' end. A modified base will be added. The 5' cap has an important function. It is absolutely required for the mRNA to leave the nucleus. So remember, in eukaryotes, the mRNA will have to go out into the cytosol in order to be translated. And once it's out in the cytosol, the ribosome will bind to that 5' cap. So 2 important functions needed to exit the nucleus and for the ribosome to bind to. The 3' poly A tail is added at the 3' end, and the name 3' poly A tail tells you exactly what it is. It's a bunch of A's added to the end. The A's are not in the gene, they're not in the pre-mRNA, they're just added onto the mature mRNA. The reason why they're there is kind of analogous to telomeres. These will slowly get chewed off by enzymes that are in the cell. So we have a little bit of space for those to get chewed on before we get to our actual mRNA. So this is going to increase the stability and lifespan of the RNA in the cytosol because, again, these will always get chewed on and these will get chewed up before it hurts the actual mRNA. So again, in RNA processing only occurs in eukaryotes. It includes splicing, a 5' cap, and a 3' poly A tail. Once that's complete, you have a mature mRNA that's now ready to be translated by the ribosomes in the cytosol.